Hello, my name is Bethan and I'm standing on a playing field in Totnes in November. Now whether you're in Key Stage 3, 4 or 5, the chances are you'll do some field work on a playing field or other grassy areas in your school grounds at some point. Now from a distance, it looks like this is just grass, but once you get up close you'll see there's lots of other plant species too. So you can see here we have ribwort plantain, creeping buttercup, dandelion and clover. If you have a really good look, you might notice that the species on your field are not evenly distributed. And that's because the environmental conditions vary across the field and plants have different adaptations or levels of tolerance for different environmental factors. The first challenge these plants must contend with is being pounded by hundreds of feet on a regular basis, which can damage plant tissue and compact soil. And you'll notice that this human traffic is worse in certain places, by the goalposts where people cross the field or where they congregate. Next is the regular mowing that the plants are subjected to in order to maintain the sports pitch. Often this mowing is less frequent around the borders of the field, so the grass is longer there. Then there are the typical abiotic factors to consider, such as sun, shade, moisture and wind. So how might we design an experiment to investigate how the abundance of one or more plant species varies with a named environmental factor? Pause the video and have a think or discuss with your neighbour now. So how did you get on? Hopefully you mentioned the need to come up with a hypothesis which encompasses your focal species and the environmental factor. And the best way to come up with a good hypothesis is through observation and some research. For example, my observations might show me the plant species looks different in the well-trampled areas to less trampled areas, or that species look different in the shady areas along the hedge to the unshaded areas. The research I might do might be looking at these Field Study Council identification guides to familiarise myself with the species. And remember, you want your non-experimental variables to be as comparable as possible across your sampling sites. You might have a problem investigating the impact of trampling, for example, if all your trampled areas also happen to be in the open area and all your untrampled areas are in the shady parts. But this can be a compromise depending on the field sites available to you. In terms of your focal species, you might want to record all the plant species present, or you might want to focus on one or two species. Those that observation suggests vary across the area, or that have particularly interesting adaptations. Next, you want to work out if you're measuring environmental change across a gradient or comparing different areas. Examples of measuring across a gradient include from a shady hedge to the middle of the grassland or from the goalposts to the field margins. Examples of comparing different areas include comparing the playing field, which is mown regularly, to areas that are left long for wildlife. Whichever you opt for, you probably want to use these useful pieces of equipment. First, a quadrat, which is just a square of a set size. This one is 0.25 metres squared, and it isolates a unit of vegetation for measuring. The reason we need a quadrat is we can't sample the whole field, so we need to come up with a strategy for capturing a representative sample. This quadrat is made from PVC plumbing pipe and connectors. It's cheap to make, sturdy and light. The next is a 30-metre tape measure, although you can use a ball of string and your feet as an alternative. Grid quadrats can also be helpful, but aren't so good for long grass, as they will squash the grass. You'll also need to have an idea of how many quadrat samples you're going to do. And the best way of figuring this out is to do one sample, see how long it takes, and then work out how many to aim for in the time available. If you're comparing two different areas, for example, regularly mown grass with infrequently mown grass, you need a different strategy. First, you need to figure out which area falls into which category. It can be helpful to produce a sketch map showing your different areas. Then you need to take a random sample in the different areas. Pause the video and have a think about how you might go about that. How did you get on? The easiest approach is to place two tape measures across your area at right angles to each other, which you will then treat like the X and Y axes of a graph. Produce some random numbers for your XY coordinates. You could write numbers on little pieces of paper, put them in a hat, and take one out without looking. Or you can use a computer to produce them. For example, I use this website, randomizer.org. When you have one number for your x-axis and one number for your y-axis, you can make a start. For example, if I have 6 and 2, I walk 6 metres across my horizontal tape measure, then 2 metres up my vertical tape measure, and that is my sampling point where I will place the quadrat. Try to make sure your range of random numbers correspond approximately to the highest numbers on your tape measure. So if the horizontal tape measure, i.e. the x-axis, goes up to 15, number your pieces of paper 1 to 15. If your y-axis goes to 20, do the same for that. If you get numbers that go off the end of your tape measure, just reject that number and pick another one using your random method. You've probably noticed I've not given you any definitions in this video, so now I have a task for you. If you're studying GCSE, find an example of an independent variable and a dependent variable from the examples in the video. A-level students can find examples of the 3 main types of sampling: random, stratified and systematic from the video. Good luck with your investigations. Let us know how you get on. The next video will tell you about some of the interesting adaptations of grassland plants to environment and human impacts on grassland. Goodbye.